In genetics, Drosophila has been used extensively to understand different genetic concepts. So this is one of the first model organism that has been used for different aspect of biological sciences. Genetics has been the front runner. Drosophila has different, very visible and distinct phenotypes. So when we say gene gives a clear phenotype, so looking at the phenotype, we can understand what is the genotype. So if two Drosophila flies are distinctly different in their phenotypes, we know that there must be some underlying genotypic difference in genotypes. Same as Mendel's experiment with pea, round and wrinkle pea or yellow and green pea. So Morgan used Drosophila as model organism to understand the order of the gene, where these genes are present, in which chromosome, what is the order of gene, and what are the genetic distance between two genes. These are important to really characterize, to identify, to understand how genes are inherited, how recombinations are happening, what are the chance of recombination between two different genes, because two different genes are giving rise to two different traits. So one of the trait, like X-linked trait, that means the trait governing gene is present in X chromosome and that is not present in Y chromosome. Okay. Two examples are like eye color and wing shape, or the wing type. So whenever we mention about linkage mapping, talking about linkage mapping, we'll be using a wild type allele and a you can say a mutant type allele. Okay. So these two phenotype, eye color and wing shape, are governed by gene M and W. So M plus or only plus will denote wild type normal wing, and small m will denote mutant type miniature wing. You can see the picture below. The left hand side is M plus or plus, that is having a longer wing, normal wing shape, and the mutant type Drosophila is having a very miniature wing, a short wing. It's a mutant type. W plus or only plus will denote wild type red eye, and small w will tell you about mutant type white eye. So you can see left hand side picture, it's having white eye, that's a mutant type eye, and a wild type wing, normal shaped wing. So the alleles are small w and m plus. W and M plus. In both the chromosome, they are having W and M plus. So W means mutant type white eye and M plus means wild type normal wing. So these are all female. So female has two X chromosomes. So that is why no genes are present in both the chromosomes. In male, they have one X and one Y. So Y chromosome, they do not have those genes. They only have one X chromosome, and that X chromosome is having wild type W allele. That's why it's a red eye, and mutant type wing, that is a miniature wing. So it's very clear from this picture. They are now crossed with each other. So what will come up next? So that will tell you whether there is a recombination or not. Let's see. In the next picture, there are two cross experiments, cross A and cross B. First talk about the cross type A. So these are the two parental type, W and plus. Plus means M plus. Okay, plus means M plus, because these are in two different loci. That's why names are given differently, W and plus. Uh, so these are white eye female, right? So that is why they have two chromosomes, two X chromosomes, because these are X-linked genes present in X chromosome, and they are crossed with miniature wing male with red eye. The same picture that is that I have shown in this slide. After crossing in F1 generation, a mutant allele in homozygous chromosomes. Right? So W plus, this W plus and plus M. So now this confirmation happens because one X is coming from mother and another X is coming from the father, from the male, and one from the female. And in male, that W and M, this is a male counterpart. Okay, so in F1 generation, in all the females of F1 generation, this will be the genetic confirmation, this will be the allelic confirmation. Okay, W plus and plus M. Okay, let's suppose this is the confirmation, because both the alleles from female are same, W plus, and they are crossed with a male with mutant type, W and M. Wild type allele is not present in this male. Okay, so these are the progeny F1 from parental type, from two parents. Now in F2 generation, after crossing of F1 with a mutant type male, will give rise to four different type of males. So one pair of the chromosome is Y, so forget about it, don't count it. So just count in that first example, in this F2 panel, white eye normal wing. Okay, white eye normal wing. So this is one of the parental type. You look at the parental type, because there are two parental type, white eye normal wing female and miniature wing red eye male. So the first male in F2 generation is similar with one of the parent, white eye normal wing, W plus. Second one, red eye miniature wing, plus M, again matching with the one of the parent. The third one is red eye normal wing, and fourth one white eye miniature wing. Now these two are not present in any of the parental uh, flies. So these two are new type of flies. New type of flies are coming up in the F2 generation. Okay, so when there is a new type of allele coming in the new generation, that means there must be some kind of recombination that has happened. So in F2, the total number of progeny is 644, where 66.5% have the parental allele confirmation, and 33.5% are having a mutant type, a non-parental allele combination, a recombinant allele combination out of 664. Count the numbers, you will get the percentage differences, 66.5 and 33.5. Okay, so you have to count, you have to quantify the number of individuals coming in F2 generation. Go to the cross number 2. Okay, you can see the phenotype of the parental type of two flies crossed, and F2 type in F1 again they are having a WM and plus plus. Okay, the allele confirmations are different from cross 1. Okay, just look at the difference. And in this F2 generation, progeny 2 from cross 2 are having 37.7% have non-parental allele combination, combination which are recombinant, and 62.3% have the parental allele combination which are non-recombinant out of 1247. So again, if you need to understand, if you want to count the genetic distance, or you can say gene mapping, you have to quantify the number of individuals those are coming in F2 generation. You have to know the genetic, the genotypes, the conformation of alleles in parental type as well as the F1 type. Okay, so when you try to map the genes, these are the informations that you need to know for sure. So these counts are important, these percentage counts are important. So in this case, in these two examples, half of the numbers, so that there are four categories. Two categories are same as parent and two categories are different because recombination is happening between two non-sister chromatids. Go to the next example. Again, there are examples that you can read. You can pause the video and see how no white eye female with normal wing mated with yellow body males. So there are two different phenotypes now, yellow body and normal wing. So the phenotype differences are white body and a white eye, and yellow body and red eye. So again, there is a genetic recombination events happening, and the trans repulsion heterozygous. Okay, trans repulsion means in a same locus two different type of alleles are present. One is plus and one is mutant type. One is plus and another is mutant type. So the alleles in same locus, they are having two different type of allele. One mutant type, one wild type. For Y or for M, look at the F1 of cross 1. Okay, all the females are having the trans or the repulsion heterozygous. Look at the cross 2 right hand side, the cis coupling heterozygous. Coupling means in same chromosome both the wild type and in the same chromosome both the mutant type alleles are present. Okay, so no matter whether these are repulsion type or coupling type, you will always get a mutant type confirmation and parental type confirmation. Okay, so in this example, cross 1 and cross 2, there was a question at that point of time whether two type of condition, whether it is trans or cis or maybe repulsion or coupling type of heterozygous, whether we'll be getting mutant type as it is or not. So in this previous example, we have seen mutant types. Okay, mutant types we have seen. So they have done another experiment to find out whether repulsion and coupling type of heterozygous can give rise to similar type of outcome or not. They have found exactly the same. So if you look at cross A F2 generation, most of the individuals are parental type and a lesser number of individuals are mutant type. So in this 1.4% are the mutant type, recombinant type I meant to say, and 98.6% are the non-recombinant type. So this percentage are saying some very crucial information. So what is this? So gene mapping and recombination frequencies are very important. So when we say these are the frequencies of recombination, this 1.4 in this example cross 1 and cross 2, the example is 1.2%. So these are the frequencies of recombination because these are the frequencies of recombinant organism. So what does it say? It says the unit of distance in genetic map is called map unit. One map unit is equal to 1% recombination. 1% recombination equal to 1 map unit. For example, two genes that recombine with a frequency of 3.1% are said to be located 3.1 map unit apart. One map unit is called a centimorgan because the Morgan actually give this kind of definition after gene mapping experiments. So they are called centimorgan. So centimorgan is equal to map unit. A distance of 3.1 map unit therefore equals to 3.1 centimorgans, and indices indicates 3.1% recombination between the genes. So 1% recombination equal to 1 recombinant in 100 crossover events. or frequency of recombinant 1%. Okay, look at the previous example. This is the frequency of recombination 1.4% or 1.2%. These are the frequencies of recombination. That means the distance between these two genes must be 1.4 and 1.2 centimorgan. Okay, so this is an example of single crossover in the lower panel where you can see the rearrangement of genes. Okay, look at this example now. Look at this example. There are two phenotypes. One is white-eyed female, white body and a normal size fly. Another is diminutive males. Okay, the male body size is small, wing size is same, and red eye. So there are two alleles. Okay, W and DM. Okay, DM. So the female and male are met, and F1 generation they have a trans heterozygous. Right, trans heterozygous. So both homologous chromosome are having both wild type and mutant type allele, W plus and plus DM. So the progeny, in that progeny, they have found in 1,000 progenies, 96.9% progenies are showing the same phenotype as parental type. Okay, we say parental type genotypes or the parental type phenotype, I meant to say. Okay, W plus and plus DM. Both these conformation can be found in the parental type, and both the phenotypes can be found in the parental type. So 96.9% are the non-recombinant type, or the parental type, and 31 individual out of 1,000 are recombinant type or the non-parental type. So something new has come up. Okay, so the percentage is 3.1%. So it says us, look at this box, the genetic distance is frequency of the recombination, that is 0.031. This is the frequency. Okay, out of 1. 3.1 is out of 100. 0.031 is out of 1. This is the frequency. So the percentage recombination is 3.1. Okay, out of 1,000 you can find 31. So obviously the percentage recombination is 3.1%. So the map distance in map unit will be 3.1 map unit or 3.1 centimorgan. So if any experiment, if you see these are the outcomes of recombinant type, you can find out the percentage out of all the progeny. That percentage will tell you what is the map unit exactly. So if the percentage is 3.1, the map unit is 3.1 map unit or 3.1 centimorgan. So we look at this figure B. We show the map units in this kind of in this way. So we write W and DM, two genes, and we show that the genetic distance between two genes are 3.1 centimorgan. In this way, we can calculate how genetic distance can be calculated. In the next talk, I will tell you how the genetic distance and the order of gene can be calculated when there are 3 genes. In this case, there are 2 genes only. When there are 3 genes, you have to understand what is the what are the combination of genes, what are the order of genes, and what are the distance between 3 different genes.